All right, we are on lab 3, cell or cytology. So let me share my screen. Okay, so here we are, lab 3, cytology or the cell. The first thing we're going to do is identify the different organelles. Some of them are going to be on models that we can easily, some not so much. And then we're going to have match up the functions to those particular cell structures. Then we're going to move on to processes and the cell structures or organelles associated with protein synthesis, and we'll discuss that. And then we'll move on to interphase, mitosis, and cell cycle. And then further to the plasma membrane, identify some features again about phospholipids. And then we're going to talk about, I don't think there are any pictures on the course page, but I'll take a look-see. We're going to discuss the effect of different tonicities of solutions on cell sizes by looking at eggs, essentially, and what effect these different caro syrup, distilled water, or egg whites have on these raw eggs. And that has to do with, from lecture, whether cells will remain the same size, will they crenate, will they get large, until they finally rupture or lyse. But that's what the last chapter is referring to. Okay, so let's start here with our organelles and cell structures, and we're going to be applying a lot of this to the models, the composite cell models that we have pictures of. So we have plasma membrane, cytosol, nucleus, nuclear envelope, 2 little asterisks. When you have the double asterisks, it means it cannot be found on the cell model. At least for me, I'm not going to ask you to try to identify those on the cell model, right? But you do have to know the functions, right? So we have mitochondria and so forth. Smooth endoplasmic reticulum, we're not going to see an example. And a vesicle, there are, I think one of the models has a number 7 that they call a little vesicle, but it almost looks like a mitochondria on end. And the other possible vesicle, is actually the lysosome. It's the only what I call a little blue jelly bean, in there. So vesicle is just a general term for a sac storing something within it. That sac is composed also of a phospholipid bilayer, but it's sort of like a vesicle. It's kind of irrelevant as what's inside. We're just saying, hey, it's like a little balloon there. If we know what's inside of it, like digestive enzymes, then we call it a lysosome. Or if we know we have more detoxifying substances like oxidases and catalases, then we call it a peroxisome. But it's sort of a general term. So things with the 2 asterisks, I'm not going to have you identify it in the model, but you do have to know the function. Okay, so as usual, it's generally easier to start with the function and match it to the organelle rather than read A through M there to try to figure out, which one goes with the plasma membrane. Okay, so let's start with A, regulates the passage of material between the nucleus and the cytoplasm. All right, so that is your nuclear envelope or nuclear membrane. So A goes with nuclear membrane. B, transports polypeptides, proteins to the Golgi apparatus for modification and packaging. So those terms, we love this. Modification and packaging is going to be the rough endoplasmic reticulum. The modification and packaging is occurring in the Golgi apparatus, though. But when they say transporting to the Golgi apparatus for modification and packaging, right, then we're going rough RER to the Golgi apparatus. Okay, C, the fluid medium in which cell reactions occur. That is the cytosol. D, contains digestive enzymes. That would be the lysosome. E, amino acid assembly. That would be the ribosome. F, produces ATP through aerobic respiration. That would be the mitochondria. G, site of DNA and RNA production. That would be the nucleus. And maybe, where we store genetic material, is technically in the DNA, but it's located in the nucleus. H, modifies and packages proteins. That would be the Golgi apparatus. I, contains genetic instructions for the cell. That would be the chromatin, because chromatin is relaxed DNA. J, synthesizes lipid, cholesterol, and steroids and detoxifies drugs in the liver. That would be the smooth endoplasmic reticulum. K, regulates transport of substances between the cell and the outside, and the external environment, or the extracellular region. Right, that would be the plasma membrane. L, the substance produced by endocytosis or exocytosis, stored in a phospholipid bilayer. That's the vesicle. So it's rather general. It's just saying that, that it's storing a substance in there. And we produce those vesicles either through endocytosis or exocytosis. And then lastly, M, anchors mitotic spindles during mitosis. Those would be the centrioles. Okay, so we're going to move on to protein synthesis. We know that protein synthesis is actually quite complicated. Protein synthesis, requires multiple organelles going through, different processes of the synthesis to ultimately come up with some kind of a product. Now protein synthesis are making different proteins. And sometimes the proteins are going to replace some of the receptors on the cell surface. Sometimes you're making, you're a cell that's making a protein-based hormone. So we're going to have a hormone, in this vesicle, and that's going to be dragged to the surface and exocytotically released. On the most part, whatever you're making is going to be dragged around the cell once you have that vesicle form, and you're going to let it incorporate into your plasma membrane. White blood cells will use them in a different way, but we're really using, we really have lysosomes, and those lysosomes will merge with engulfed bacteria and viruses and things. Okay, so we have the processes: transcription, modification, and packaging, exocytosis, translation, replication. Those are in bold print. Those are the terms, and only those terms, to be put in where we have process blank line, right, all the way down in this chart. And the cell structures you get to choose from, because it's not going to be just anything in the cell. Golgi apparatus, nucleus, ribosomes, vesicles, vesicles, backslash plasma membrane. Blech. Okay, so in protein synthesis, if we start with DNA, we end with DNA, because you need to be able to replicate your DNA to really make anything. Right? That process is called replication, and the structure in which it occurs is the nucleus. Remember, DNA stays in the nucleus unless the cell is divided. And then we're going to let it, going to condense it and split, drag it into the middle and split it apart so each cell gets a copy of the DNA. All right, so DNA to DNA is replication, or you can put DNA replication, the structure nucleus. All right, but when we are making proteins, we need to get a copy of the DNA to go out into the cytoplasm, and DNA doesn't leave the nucleus unless the cell's dividing. So we have to have the photocopy form, so to speak, to go out to the ribosomes, and that's what the RNA is, more specifically ribosome messenger RNA. All right, so when we start with DNA and we end with RNA, that process is called transcription, and that occurs in the nucleus as well. So transcribing means to write it down. So we always, thought of it as, well, I'm going to write down the recipe for that protein, the directions from DNA to RNA. We're going to write it down in transcription. All right, the RNA can leave the nucleus, and now it goes out into the cytosol, where we find ribosomes. So now that the RNA is out there, what organelle is going to produce that long amino acid assembled chain like a polypeptide? That process is called translation, and the structure of the ribosomes. As we said, ribosomes function as, in amino acid assembly. They're going to take the information from that messenger RNA, And essentially, with the help of what's called transfer RNA, is it's going to produce the sequence of amino acids for that initial sequence for this protein. All right, so that process is called translation, because we're going to now translate it from a written form to a thing in the ribosomes. Well, now we have that polypeptide, and how do we convert that into the functional protein? Because remember, we often have to go, or we do, go from a primary to a secondary to a tertiary, and even maybe a quaternary structure. And all held together by these hydrogen bonds that maintain those fancy shapes, and that's what gives it specific functions. So that process is called modification and packaging, and the structure is the Golgi apparatus or the Golgi body. All right, now we have now packaged it. It's going to bud off in the form of a vesicle because we have this functional protein now. Let's say it's albumin or something, and we're going to, and it's from there to the extracellular fluid. So from within the cell in that little vesicle to outside the cell. That process is going to be exocytosis. And the structure would be the vesicle's plasma membrane. Really, I'm only going to be able to point to a plasma membrane. Okay, so how do I ask these questions? And when we look at the images, you'll have a better idea. But, since we just looked at the Golgi apparatus, nucleus, ribosomes, and the plasma membrane in the previous exercise, because we're going to apply those to just, identifying those organelles in a cell. Right, and like, what is its function? Let's say it's a ribosome. You're going to say, well, amino acid assembly. But now I can add another layer to it. If I point to a ribosome and I ask you, what process of protein synthesis does the ribosome impart? Or maybe I'll point to a ribosome and say, what process of protein synthesis, process of protein synthesis from this second exercise, we're talking about these processes, does this, and I'm pointing to a ribosome, does this organelle impart, or, something like that. So I'm looking for modification and packaging, or modification. If I'm pointing to the nucleus and I ask you what, process of protein synthesis or, occurs in this organelle, and I'm pointing to the nucleus, the big blue ball there, there's actually two things, right? We have replication and we have transcription occurring there. So you might have transcription, modification and packaging, replication, more than one above, or it might just be transcription or replication. Just read it. They'll either have an option to have both of those things because two things do occur in the nucleus, or it might just be one, and given your options, only one of those legitimately occurs in the nucleus. But we'll look at that again when we get to looking at the images. We'll revisit this a little bit. But the fact that we have these listed as processes here and then the cell structures, I'm going to ask you like that. What process of protein synthesis? Now I can point to the plasma membrane. What process of protein synthesis, does this organelle, contribute, or something like that? I forgot how I phrased it. And you would say exocytosis. Okay, now we're going to look at interphase and mitosis and the cell cycle. So as you probably know from lecture, the cell cycle are all the steps the cell goes through when it needs to replicate itself. Right? Interphase is interphase. Certain things happen in interphase. And mitosis means, the steps associated with a division of the nucleus. And those mitotic phases are prophase, metaphase, anaphase, and telophase. And then at the end of mitosis, cytokinesis is complete, because cytokinesis is division of the cytosol, and all the organelles. Okay, so mitosis phases set up, identify, because it's on the side of the room, identify the phases from the slides and the models, because they're generally on one side of the room, but we're going to show you what you need to know on this online version. Identify centrioles, chromosomes, spindle fibers, or what's called mitotic spindles, cleavage furrow on the models, and identify where cytokinesis is complete. All right. So we have models, what we call cell on stick models. And then we have these large, I call them the cookie sheet models. Looks like a great big cookie sheet, but it's, it has all these diagrams, so to speak, of the cell going through these different phases. And so even on those, it's easy to identify what the chromosomes are or the centrioles are and what the chromosomes are and the mitotic spindles and so forth. Okay, so what we're first going to do is look at each phase and put together what is functionally occurring there. All right, so it says match the functions to the phase and apply to the model slides anything that you have that is representative of interphase, prophase, metaphase, anaphase, or telophase. Okay, so here are our options. Chromosomes line up in the middle, DNA replication and protein synthesis, so forth and so on. As usual, the options to go in these fill in the blanks are in bold print. So let's start with interphase. What occurs during interphase? And you can print these off and write them out by hand. If you're downloading the document, and you can put it in like an edit form, then you can copy and paste maybe into it, but otherwise I would print them out and write it out myself. DNA replication and protein synthesis go with interphase. Okay. DNA replication and protein synthesis. Prophase, 3 things occur. Boom, boom, boom. We have, and I'll go to the top, here. The first thing that we would put in there is chromosomes coil. Out of this list, I'm just kind of going down the list, left side to the right side. Chromosomes coil. Centrioles migrate to the poles and mitotic spindles are produced. That also occurs in prophase. And the nuclear envelope breaks down. That's the third thing. All right. In metaphase, chromosomes line up in the middle. All right. Chromosomes line up in the middle. Where is that? Oh, it's our first option over on the left-hand side. There's kind of a way you can think of this. Metaphase is in the middle of these 5, and metaphase is the middle stage, and chromosomes line up in the middle. Or they actually line up in a linear orientation. The mitotic spindles drag them to the middle, and then we're going to pull them apart. So in anaphase, chromosomes pull apart, and the cleavage furrow begins. Right, cleavage furrow right here, 3, third option on the left-hand side, cleavage furrow begins. The beginning of that cleavage furrow, or the cleavage furrow beginning, marks the beginning of cytokinesis, because as the chromosomes are getting pulled apart, we start trying to pull the sides of the plasma membrane of that cell toward one another in the center, so it will pop into 2 cells. So we actually start that process in late anaphase. Okay, telophase. The 3 things that occur in prophase, opposite are going to happen in telophase, or the 3 things that happen in telophase are the opposite 3 things that happen in prophase. I guess that's articulated a little better. Okay, so we have the nuclear envelope and membrane starts to reappear, because now that we pulled the chromosomes apart, we don't want those chromosomes or the mitotic spindles to let go of them and have the chromosomes just kind of float away out into the cytosol. So you very quickly start to remembrane bound them, so we contain the DNA, in the nucleus. So the nuclear membrane, reappears and starts reappearing. Chromosomes start to relax, and the mitotic spindles break down. So the mitotic spindles are going to let go of those chromosomes now that we're starting to remembrane bound them, and the chromosomes will start to relax. And then lastly, cytokinesis is now complete. So toward the end of this, now that we're remembrane bounding, the nucleus, because that's what mitosis is, division of the nucleus. Now we've divided it, and we're now establishing another nuclear membrane around them. Now we have to complete the cell division, the rest of the cytoplasm, or, the cytosol and the organelles. And then we have 2 cells, and thus we end with daughter cells. Now, daughter cells are not a phase, but the product of telophase and cytokinesis is our daughter cells. All right, we'll look at models in a moment. The plasma membrane. Use the model of the phospholipid bilayer to identify the phosphate groups, the fatty acid tails, which we actually looked at in the chemistry, right? But we're going to look at that again. Hopefully there's going to be another picture in there. I can't remember. Often I get to this point and I either have to go back to the other image and copy it over, and hopefully it'll be in there. But when we looked at it, and we had that model, we have the white ball and the two little red tails. That's one phospholipid. Okay, remember phospholipid, you go to the chemistry chapter, right? Go, well, what are the building blocks? One glycerol, two fatty acids, and one phosphate group. And we said that in that phospholipid, we had both, it was the combo pack of bond types. Some regions are polar covalently bonded, others are non-polar covalently bonded. So now we're asking you here, identify the bond type of the phosphate group. That's the polar covalently bonded end of the molecule. Identify the bond type of the fatty acids, non-polar covalently bonded. And this should be reviewed from the chemistry chapter in the previous week. Okay, and this information we actually covered in there too. Which part of the phospholipid bilayer demonstrates hydrophilic behavior? So hydrophilic means liking water. So that would be the phosphate group, because phosphate group is polar covalently bonded, water's polar covalently bonded. Boom. So what is the behavior of the phosphate group with water? It's hydrophilic. So there's different ways to ask the same question. Which part of the phospholipid bilayer demonstrates hydrophilic behavior? The phosphate group. Which part of the phospholipid layer demonstrates hydrophobic behavior? Those would be the fatty acids. Okay. Now we're going to move on to the tonicities of solutions and their effect on, cells of the body. Here we're looking at eggs. So raw eggs were added to different solutions to observe water movement through a plasma membrane. All eggs were added to a vinegar solution first, and afterwards they are added to one of these three solutions listed below. Observe the eggs and answer the questions. So I'll talk about this and I'll tell you, what they look like, because I don't think we have pictures of the eggs on the course page. But these are really theory questions, and I wouldn't have shown you pictures of the eggs from the lab for testing purposes, but I might ask you what would be the effect of these different tonicities on a cell. But let's just get through filling in this chart first. So we have solution types over the left-hand column: Caro syrup, distilled water, and your egg whites. Those are our three different beakers, so to speak, filled, with these different solutions. And then you're to identify the tonicity of the solution. Will it be considered hypertonic, hypotonic, or isotonic? Will I give you this information? Right? The Caro syrup is the hypertonic solution. Distilled water, not saline, right? This is distilled water. That is a hypotonic solution. And the egg white is an isotonic solution. It acts as an isotonic solution. So we look at the eggs in these environments, right? And then we go, okay, what did we observe? Do the eggs stay the same size? Do they shrink or crenate, or do they get enlarged and maybe lyse, and break somehow? So the hypertonic solution, the observation, what you notice that cell is that the egg shrunk, and it does. It shrinks and it gets smaller and more condensed. In the distilled water, the hypotonic solution, the egg got much larger and kind of blown up like a little balloon. And the egg white isotonic solution made no difference in the size of the egg. And that's what we would expect from red blood cells or different body cells, especially in blood, in plasma. If it's acting as a hypertonic solution, it draws fluid and water osmotically out of the cells, and those cells start to shrink. In a hypotonic solution, it's the opposite. If your plasma is too hypotonic, then water in higher concentration outside the cell migrates into the cell, or I should say osmotically drawn, and the cell starts to get larger, like you're blowing up a balloon. And it may get to the point where it actually ruptures. In an isotonic solution, it makes no net cell size or egg size difference. The ratio of solute to solvent outside is the same as inside. So there's no osmolarity, there's no osmotic gradient, there's no profound difference between the concentration of water outside the cell in the solution the cell or the egg is sitting in compared to inside the cell. So you get a little bit of water movement, into and across the membrane through those aquaporins and stuff, but very little, and it makes no demonstrative change in the cell or egg size in this example. Okay, so what effect did the Caro syrup have on the egg? It caused it to crenate. Why? Because the hypertonic solution, draws fluid, water from where it's in high concentration inside the cell to out of the cell, and the cell shrinks. What effect did the distilled water have on the egg? It caused it to get larger because water was moving from outside the cell in the hypotonic solution into the cell. That's your osmotic gradient, is into the cell. And so it makes it enlarge like you're blowing up a balloon. And what effect did the egg white have on the egg? None, because it's the same tonicity inside the cell as outside the cell. Okay, so what I'm going to do is page up, and let's look at some of the images. Let's go to 3. Let me shrink. Oh, no, already left it. Okay, so we're going to start at the beginning. Remember, not all these images are necessarily going to be relevant. Am I going to use them? But we're going to begin by looking at this, what's called the composite cell model. And we're going to do the best we can to identify the organelles that are easy to find, and which we looked at, we identified functions for in the first lesson. And remember, some of those organelles had 2 asterisks, so we're not going to find them, very readily on the models here. Okay, so this is actually we can see a number of different organelles here. I will tell you right now is that this big blue ball in the center here, that's the nucleus. And the little white dots scattered throughout, that is the chromatin, the relaxed DNA in the nucleus. Now, this is a nucleolus, this little pink ball with the blue dots on it. You don't have to know it, okay? I mean, people always ask me, well, what about, is that the nucleus? No, that's the nucleolus. That's where ribosomes are formed. But you just have to know the big blue ball in the center, the nucleus, and the little white dots there, the chromatin. Very quickly, I'm just going to point to you. See, they look like red jelly beans here. You can see the outer surface. Below that at 3 o'clock, you're cutting, you can kind of see into the surface. These big red jelly beans, right, those are the mitochondria. Over here, about 9 o'clock, number 11, those blue sticks, those are the centrosomes, or the centrioles. Let's see, what else can we see quite readily? Oh, over here, number 9, that's the blue jelly bean. That's going to be a lysosome. By about 5:30, 6 o'clock, you can see part of these brown stacks of sacs. That's going to be your Golgi apparatus. These yellow orangey layers with the dots on the surface. And then down here further, like number 5 is pointing to these. These are those same sacs that have ribosomes on the surface. It's just cut in a different direction. Those are all rough endoplasmic reticulum. And look how abundant it is all over the cell. I mean, we have smooth endoplasmic reticulum, but there isn't a good example on here of that. And that is much less numerous. Then look at all the rough endoplasmic reticulum. They look like little centipedes. All right, so that's what I can pick out right away and point to for you. Okay, so here he gave you kind of a numbered, if you can go back that page and look at what number 1, the plasma membrane is and so forth. We're going to show you that later because he goes, it's later. The nucleus is the big blue ball. The little white dots in that are the chromatin. the rough endoplasmic reticulum, they kind of, it uses number 5, but it is quite abundant. They kind of look like caterpillars. The ribosomes are the dots, or the studs on the surface of that rough endoplasmic reticulum. And then the centrioles were number 11. They anchor mitotic spindles. Okay, so back here again, and now the camera's moved down a little bit so we can see a little bit more. Right. When he said the cytosol, or the cytosol is listed there, that's the light blue background here. I get that. Here's a number 1 here at like 3 o'clock. But the light blue background is the fluid in which it's all sitting in, and that's called the cytosol. Mostly water for chemical reactions to occur. And to also to allow these organelles to be suspended. Again, we can see the red jelly beans, rather abundant throughout. Now we can see number 10, those brown sacks of stacks. That's the Golgi body or Golgi apparatus. 11, again, over at about 9 o'clock, those blue sticks. Big nucleus, the big blue ball in the center, and the little white dots are the chromatin. This is the plasma membrane. So this model here actually sits, in this clear plastic portion, and that represents the plasma membrane. Plasma membrane. Phospholipid bilayer regulates the passage of material into and out of the cell. All right. Separates the internal and external environment, functions to regulate what comes in and out of the cell. Okay, here we're back to that model. Oh, good. And this one's a little bit better than the one we have in our lab, because ours it's kind of falling apart. But here you can see the white ball with the 2 little red tails. Right? That's one phospholipid. It's a bilayer, so there's 2 layers. Here we have the white ball and the red tails. And then if the card, which will maybe get moved, you can see the little white ball with the red tails pointing to the other red tails. And that's how these phospholipids orient themselves. That's why if you understand that the fatty acid tails are nonpolar covalently bonded, they don't want to be exposed to the water that's outside and inside the cell. So they point to each other, like oil beating up when you put it in water. And it doesn't take a lot to keep them that way, because they don't want to face outward. They want to point toward each other. So it has to do with the whole solubility, issue, where we say like dissolves with like. Fats want to dissolve with other fats, or nonpolar covalently bonded things, or things just without charge. But the phosphate group is polar covalently bonded, like water, like amino acids, like glucose, blah, blah, proteins and whatnot. So it likes to be pointing outward. It's hydrophilic. Okay, here the phosphate transport liquid soluble molecules. They're pointing to the fatty acid tails anyway. That, don't worry about it. It's supposed to be like a transport molecule, a protein. I don't ask about it. I think the phospholipid bilayer is just enough. So don't worry about the transmembrane protein. Okay, this starts to get into, I'm not sure. Oh, we're already into lab 4. Okay, so I was going to say, how did we get into the next lab? So let me back up and go back to the lab 3 images that we just looked at. All right, and that completes them. Now we have that image here, the phospholipid bilayer, the PEG. Let me just check some of these other ones. I think we should be otherwise in good shape. I think I gave you a card. I don't know why it has hyaline cartilage. So I'm going to change this, you guys, very quickly, because I think the wrong image got uploaded. So, and let me just go back. Forward. Okay. Because somehow we went right to lab 4 from there. So I'm going to go and change that real quick, right? After we finish this Zoom. So my apologies. There's another image that should have gone up there. Okay, so now we're going on to the protein synthesis stuff that we were talking about. And here we have a model of DNA and another model of DNA. And he's saying, okay, one is A and one is B. So what is the process if we go from A to B, the molecule on the left to the molecule on the right? Well, if we start with DNA and we end with DNA, that would be DNA replication or replication, right? It's one of the processes of protein synthesis. Now we start with A, which is the left-hand molecule DNA, and we end with the single helix B on the right. Start with DNA, end with RNA. That is transcription. And both of those, processes occur in the nucleus. Now we have the RNA and we produce this polypeptide here, an amino acid sequence. What process is that? That's translation. Now we read the recipe and we produce the product. In what organelle does that occur? Ribosomes. For some reason, the image stops there, but the last step would be pointing to the plasma membrane. If I pointed to the plasma membrane, or from there after you transcribe, that and you produce this polypeptide, you actually go into the Golgi apparatus for modification, and then from the Golgi apparatus, the protein buds off in a vesicle, and we drag it to the plasma membrane, and we exocytotically release it. In these, I'm not sure why those images weren't included, but let me go back a little bit to make sure that you understand what I'm talking about. So let me go back to just back up a little bit. We're talking about, we are talking about these processes of protein synthesis. And I said that, we're identifying in what organelle did they occur. And I'm going to go back to, we have one of these composite pictures. So hold on. I had to go back farther than I thought. Sorry about that. I should have just gone right back to the beginning. Okay. Back to 3-3. I should have gone back to the beginning. All right. So we're saying that, for instance, I could point to the blue ball here and I could say, what kind of question might I pose? I could point to the blue ball and just say, identify this organelle. And I have an arrow pointing to the big blue ball. And I usually will say the big blue ball or the blue ball. Right? Because I have to distinguish between the nucleus and the chromatin, and you can't tell that unless I say, the big blue ball. Right? So that would be the nucleus, right? What if I pointed to this big blue ball and I said, identify one process of protein synthesis, maybe in bold print or in quotes or something, hopefully italics, I'll try to bring your attention to it, that occurs in this organelle. Well, we know the first two things happen there, DNA replication and transcription. If I pointed to this ball and I'm pointing to the white little dots there, I could ask you, identify that organelle, and I put like the white dots. Then we're talking about the chromatin. I could point to those white dots and say, identify the function. So that type of a question, identify and give me the function, is from the first exercise. If I'm asking you about steps or processes of protein synthesis, that's exercise 2. Further down here, let's say here, number 10 is the Golgi apparatus or Golgi body. I could ask you, identify that organelle. And you would say the Golgi apparatus or Golgi body. It's multiple choice. Identify the function of that organelle at the tip of the pointer, or at the arrow or arrow A. Then, you would say modification, packaging of proteins. I could ask you, identify the process of protein synthesis that occurs there. That would be modification and packaging. Here we have the rough endoplasmic reticulum. They look like little worms, or caterpillars. So I can point to, let's say I used number 5 here, right, at about 3 o'clock, number 5. Identify that entire organelle. You would say rough endoplasmic reticulum. I could ask you, identify the function of that entire organelle. Or I could ask you, identify that structure, or no, not the structure, identify the organelle. And I'm pointing to the little ribosomes on its surface. Then I'm going to say, identify that organelle, the dots. And because the dots on the RER are the ribosomes. As a unit, that sac and the ribosomes on the surface are the rough endoplasmic reticulum. So when it comes to like here on the RER and here in the nucleus, because it's hard for you to distinguish, is she asking for the nucleus or the chromatin? And on the rough endoplasmic reticulum, is she asking me about the RER as a total or maybe just the ribosomes? I will be as clear as I can. The entire structure or maybe just the dots on the RER, ribosomes. And in which case, if I was pointing to the ribosomes, I could ask you, identify that organelle, and you would say ribosome. I could ask you the function, amino acid assembly. I could ask you what process of protein synthesis, does it participate in, or whatever. Then you would say translation. So hopefully you get that concept. And I kind of apologize in advance that we don't have images that follow in that sequence, and when we get down farther in the protein synthesis stuff, but not all instructors go all the way through, and I think they make really easy questions, so I go all the way through. Plasma membrane, right? I could point to the plasma membrane. Let me just back up. Say, identify that organelle, and you would say plasma membrane. And this is from lesson 1. Identify the function of this organelle. At the tip of the pointer or something, or arrow A, or whatever, the arrow. You say regulates the passage of material into and out of it. It depends what the options are. Remember, it's like a multiple choice. But now I could also ask you what process of protein synthesis occurs here. And that would be exocytosis. Okay, so if you have any questions about that, just shoot me an email. We can always schedule a live little Zoom here. Here's our phospholipids. Let's get past that. Okay. We're going to get started on, I'll fix that. My apologies. Oh, no, actually, let me go back. I'm not totally crazy. It's just that they're all in one. All right. And they were scanned together and I couldn't separate them. But this is a cute little picture of the phospholipid bilayer. So I'm pointing to, I draw out the phosphate group. Here's your hydrophilic head, your hydrophobic tails. The hydrophilic head are the phosphate groups, polar covalently bonded. The hydrophobic, region, the tails are the fatty acid tails, non-polar covalently bonded, and pointing to those. I still may try to reorganize this somehow so that the phospholipid image is on top. Okay, keep moving so that this doesn't take too long. Sorry, you guys. All right, so here we have replication, transcription, translation, and then for some reason we just dropped the ball there. But now you understand how I might, pose questions and how I do pose questions, actually. Okay, this takes us to the phases, interphase, and our mitosis phases, prophase, metaphase, anaphase, and telophase. All right, so although we have 1, 2, 3 here, you would think that those be representative of interphase, but the lower right-hand corner of this great big, what I call cookie sheet model, will be your interphase. These phases 1, 2, and 3 are prophase. All right, prophase, 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 1, 2, and 3. What occurs in prophase? The nuclear membrane starts to break down or disappears. The chromosomes coil. Centrioles or centrosomes travel to opposite ends of the cell and build the mitotic spindles. Okay, prophase, nuclear membrane, chromosomes, blah, blah. Pretty much what I say. Okay, then we go to number 4. All right, now here the chromosomes are lined up in the middle. right? So this is metaphase. Chromosomes line up in the middle. Very obvious. Those are the blue X's there. Now let me just identify two. To the outside of this cell, that brownish, darker brownish layer, that would be the cell's plasma membrane. Right. The light tannish background is like the light blue background in those other models. That would be the cytosol. You see the black dots, the pair of dots to the right, and then to the left at like 3 and 9 o'clock, I guess you'd say. Those are the centrioles. And they build the mitotic spindles, these number 10, these red spindles attached to the chromosomes, and they line them all up in the middle. Okay. Now we have to start, now that they're lined up in the middle, let's start pulling them apart. So 6, this space here with a little bit of indentation here, this is anaphase. It's a slightly late anaphase, but it's an anaphase. All right, so number 6 is an anaphase. Chromosomes are getting pulled apart. See the centrioles that have those red mitotic spindles attaching to the chromosomes. It's like reeling in a fish. They start shortening and pulling those chromosomes to one end of the cell and the other. And when they get to a certain distance, we start pulling down in the center, the plasma membrane, top and bottom, toward one another. And that is a cleavage furrow that begins late anaphase, and that marks the beginning of cytokinesis. Okay, see, cleavage furrow begins, chromosomes pull apart, cleavage furrow actually starts late anaphase, but that is the beginning of cytokinesis. All right, then lastly, we have telophase. So you see how, distant these chromosomes have now come to one another, and we're pinching in more and more. That cleavage furrow is becoming more and more exaggerated until finally the top and lower margins will meet. Okay, so this is telophase, the last stage of mitosis, nuclear division. So now the mitotic spindles are going to let go of those chromosomes. We're going to remembrane bound them so the chromosomes don't start flowing off, all over the place. And the chromosomes start to relax. We now pull the margins here. The cleavage furrow becomes more pronounced, and it pinches off into 2 cells. This represents telophase. And once we really get these nuclei nicely well established here, we will finish pinching off in the center, and cytokinesis is complete. All right. Cytokinesis will be complete. It's just the opposite of prophase, which is what it does. Now we have the 2 cells. These are the 2 daughter cells from the original cell. We copied all the chromosomes, the DNA, all the organelles, and now we have 2 identical cells. So this really isn't a phase. Technically, daughter cells is not a phase. However, we use this as representative interface because once those chromosomes, let me just go back to telophase. Once we remembrane bound these and these chromosomes have gotten all loosey-goosey again, and now they're actually loose enough to start replicating again, we go right into interphase. It starts to replicate its DNA again. The cell starts to, again, divide in half. All right, so here we have the progeny, the 2 daughter cells of this process, and they go right into, they enter into and cycle back into interphase. So this phase number 8 here with the 2 cells, right, we use that as an interface. So I could point to this and it's identify this phase, and you would say interphase. Or I could point to this and say, identify, what is functionally occurring in this phase. Protein synthesis, DNA replication, something along those lines. Further, if we went back and we, that's how I would ask questions about these phases. I would say, okay, identify this phase, telophase. Identify what's functionally occurring or a function that's occurring here. And, this is going to be a multiple choice. So, there'll be one, there'll either be one complete answer or it might be A and B, something like that. Okay. And so that's how we use those models. Here are examples of what we call cell-on-stick models. We usually have these placed next to the microscopes, and the microscopes are each one pointing to one of these phases. We have from right to left, interphase, prophase, metaphase right in the middle, chromosomes line up in the middle. Anaphase, chromosomes start to pull apart, and we start to see the cleavage furrow. Telophase over here, this is great. It really pinches in. We don't have two cells quite yet, but we pulled them way far apart. We're going to start remembrane bounding these, and the chromosomes relax, and the mitotic spindles let go, and then we would end with two new cells. Interphase, prophase, metaphase, anaphase, telophase. That's on the cell-on-the-stick models. These just kind of help you see what they would look like microscopically. Here are the chromosomes. I mean, I like the anaphase. I love the telophase in the lower right-hand corner. But we would start in the upper left as an interphase. And that nucleus staining that dark is actually questionably, like, I think I would look for a version that you can see the nuclear membrane, and it's not quite so dark stained because the chromosomes are replicating, and they're not as dark staining and condensed. But in the next picture, prophase, they definitely are darker staining. So, I think it's better. All right, so that's prophase, and then we see the metaphase, chromosomes line up in the middle. Anaphase, the little chromosomes are getting pulled apart. And telophase, you can just see where it's really pinching in, and some mitotic spindles are here right in the center. But we just about have two cells. This represents the response of the eggs. We have A, B, and C, the different regions. We used to use potatoes, and now we use eggs. So it just goes to say, hey, if A is the hypertonic solution, B is the isotonic, and C is the hypotonic, then you can anticipate what effect it would have on the potatoes. Hypertonic solutions always cause something to shrink because it sucks the water out of them. Hypotonic solutions always cause stimulate a cell or the potato or the egg to blow up like a balloon and enlarge. And the isotonic solutions have no effect on the cell size, no demonstrative effect. I mean, little bit of change in size, but nothing demonstrative. So the cell or the potato or the egg would remain unchanged in an isotonic solution. Okay, and that is the end of this lab.